Welcome to our celebration of the 125th anniversary of the Journal of Experimental Medicine.
Since 1896, JEM has been a leader in publishing studies in immunology, cancer biology, vascular biology, microbial pathogenesis, neuroscience, and stem cell biology.
Throughout its history, JEM has championed studies that have become landmarks in the field of biomedicine.
Thanks to the high quality of studies, JEM is at the forefront of scientific publishing while remaining a not-for-profit outlet.
JEM represents the home where many leading scientists found their passion for science by reading about other people's discoveries.
We would like to take this opportunity to acknowledge the authors, reviewers, and readers who make JEM what it is every day.
We are thrilled that 5 prominent scientists will start off our celebration by highlighting key JEM papers. Please join us and listen to these short talks by Dr. Akiko Iwasaki from the Yale School of Medicine, Dr. Joseph Sun from Memorial Sloan Kettering Cancer Center, Dr. Gwen Randolph from the Washington University School of Medicine in St. Louis, Dr. Stephanie Eisenbarth from the Yale School of Medicine, and Dr. Ali Ellebedy from the Washington University School of Medicine in St. Louis.
You can find more JEM 125th anniversary content on our website throughout the year. Happy birthday, JEM!
Hello, my name is Akiko Iwasaki. I'm an immunologist from Yale University School of Medicine. Happy 125th anniversary, Journal of Experimental Medicine. I'm happy to share one of our most recent papers that was published in the Journal of Experimental Medicine as of January this year. And we investigated the neuroinvasive potential of SARS-CoV-2 in this paper. This work was led by Eric Song and Che Zhang. We used 3 distinct approaches to probe whether SARS-CoV-2 can infect neurons. The first model involved human brain organoid, and this was an infection that was done in vitro. A second model employed mouse model of SARS-CoV-2, where we can selectively infect animals in the brain or in the lung to study the consequence of such localized infections. And in the third model, we took biopsies or autopsies from humans who had lethal COVID infection to determine whether there are any cells that are in the brain infected with the SARS-CoV-2 virus. Using the first model of human brain organoid, we were able to demonstrate clearly that there are neurons in the organoid that are infected by SARS-CoV-2, as indicated in the red staining here. And what's interesting was that the infected cells were not the ones that were dying of this infection, but the bystander neurons appear to be dying of infection, as indicated in green, which are indicating apoptotic cells. And using transcriptome analyses, we demonstrated that the infected neurons are hyperoxic and have enhanced metabolic status compared to other neurons. And in fact, this might be leading to a heightened production of the virus and supporting the replication and production of the virus. Whereas the bystander neurons had hypoxic phenotype, leading to apoptosis. So this really led to the insight that neurons are becoming infected, at least in this organoid model, and that it's causing the death of nearby neurons by potentially sucking up the oxygen that's needed for the metabolic need of an infected cell to produce more virus. Moving on to the second model, where we used a mouse model in which mice can either become infected specifically in the lung or specifically in the brain. And we made these mice by introducing AAV that encodes for human ACE2, either into the lung only or the brain only, allowing us to selectively study the impact of SARS-CoV-2 infection in these organs. And what we discovered was that animals that were infected only in the lung, indicated in blue, were all survived, whereas those animals that were infected in the brain all succumbed to infection with much less viral inoculum, indicating the lethality of neurotropic infection caused by this SARS-CoV-2 virus. Finally, looking into the autopsy samples from postmortem COVID-19 patients, one of 3 patients had a striking phenotype in which we detected spike protein within the neurons of the frontal cortex in this particular patient, indicating that neurons can become infected in some cases upon COVID-19 disease. So to conclude, our paper demonstrated using 3 separate approaches that neurons do become infected with SARS-CoV-2 with brain organoid model, in vivo mouse model, and autopsy. And in vivo mouse model, we demonstrated further the lethality of neurotropic infection compared to the respiratory infection. And so clearly future studies are needed to understand neuroinvasion consequences in acute and long COVID, but we hypothesize that there might be a event happening within the brain which enables infection of the neurons, and that further leads to local hypoxia and ischemic infarcts that might compromise the blood-brain barrier in these settings. Thank you for your attention.
My name is Joseph Sun, and I'm a member in the immunology program at Memorial Sloan Kettering Cancer Center. And I'm a professor and the director of our immunology and microbial pathogenesis graduate program at Weill Cornell Medical College. My lab is interested in the general biology of natural killer cells, or NK cells, and specifically their role in infectious diseases and cancer. However, I began my scientific career by studying T cells. And along these lines, I will present the first scientific paper I was introduced to during my junior year of college at UC Berkeley. I was taking the molecular immunology course taught by Neelab Shastri at the time. Neelab recently sadly passed away. But Neelab was a fantastic teacher and one whom I credit for introducing me to immunology in such an exciting way that I knew I wanted to devote my career to understanding the immune system. That quarter, he made us read a Journal of Experimental Medicine paper from the group of Mike Bevan, and it's entitled Positive Selection of CD8 positive T cells induced by major histocompatibility complex binding peptides in fetal thymic organ culture. I actually still have the original printout from Neelab's class. That's now a little bit faded. This was a study driven by Chris Hoagquist when she was a postdoc in Mike's lab, along with a graduate student, Mark Gavin. Now, I didn't know it at the time, but both would become close friends and colleagues in the years to come. The background of this paper is that positive and negative selection had been described by various groups in the late 80s and early 90s, including Mike's group, but also Pippa Merrick's, Harold von Boehmer's, and many others. And they used a variety of systems and mouse models to study developing T cells in the thymus. Now, one of these interesting tools that was developed at the time was the FTOC, or fetal thymic organ culture, where the fetal thymic lobe of beta-2M deficient mice, now these were newly generated at the time, which lack MHC class 1 because they lose the beta-2M component. So the fetal thymic lobes from these mice could be cultured for up to 10 days in vitro, during which time exogenous beta-2M, along with peptides, could be added to drive the development and selection of thymocytes in vitro. And the FTOC was the key culture system that was used in this JEM study. In the early 90s, although evidence supported positive and negative selection of thymocytes, the molecular events that drive these selection processes in the thymus were poorly understood. So Chris and Mike's JEM study in 1993 elucidated the importance of both MHC class 1 along with specific peptides in driving thymic selection. And again, they used addition of beta-2M to these beta-2M deficient FTOX along with peptides. They showed that using various mixtures of beta-2M and peptides along with different peptide concentrations, that it was the diversity or complexity of the peptides that was more important in driving positive selection than peptide concentration. The concluding sentences of their abstract reads, "These data argue for a specific role for peptides in the process of positive selection. Furthermore, this culture system should be useful in identifying peptides that can promote positive selection of cells expressing a specific T cell receptor in TCR transgenic mice." And we know that this set the stage for Chris's Cell paper that came out the following year, first describing the generation of the OT1 mouse, which has a specific TCR for the synfecal peptide of ovalbumin that is bound by H2K of B, or MHC class 1 in the B6 mouse. It's a mouse that everyone in the field now knows and has probably used. It was this study that in part motivated me to join the University of Washington immunology program for my own PhD studies, and of course choosing Mike Bevan's lab for my thesis work.
I'm Gwen Randolph. I'm at the Washington University School of Medicine in St. Louis, and my laboratory is interested in the trafficking of immune cells and molecules from sites of inflammation to downstream locations like lymph nodes via lymphatics. And one of the cell types that we've studied is the monocyte and their accumulation in lymph nodes. So I wanted to share with you a favorite paper in Journal of Experimental Medicine that influenced me early in my career and influenced the field. And it's a paper that I still share with students when I teach about lymph node organization. So this paper came from the laboratory of Uli von Andrian. The first author is Roger Palferman, and key collaborators include Stefan Jung in the Dan Lippmann lab, Barrett Rollins, and Antal Rott. And the beauty of the paper is that it shows a new mechanism at the time of how monocytes might move into lymph nodes. We had been focusing on their trafficking through lymphatics to lymph nodes, which is an actual event that occurs, but also here we find that the inflammatory side will make chemokines, which of course are small molecules, about 20 kD. And the laboratory put together, the von Andrian laboratory put together how important that small size was to the way the lymph node was structured. So we understood already at the time that key structures in the lymph nodes were these conduits that allowed for relatively small molecules lower than 70 kD, to enter them and make their way essentially to the high endothelial venule, as you can see schematized here. So what the laboratory came to understand was that, of course, we want lymph nodes that are draining sites to be reactive to what's going on upstream. And one of the ways in which that is controlled is by molecules actually coming in to control, through their chemotactic activity, the recruitment of cells like monocytes therein. And so the data were to simply look at the kinetics of cell accumulation in lymph nodes, and then look at the proteins that would bring them in, like MCP1 for monocytes. And the realization was that there was protein in that draining lymph node, but there was essentially no increase, even at the time of maximal monocyte accumulation, of the RNA for that protein. And that's because the RNA is not made there. It's all remote. It's made in the skin, and then flows down to the lymph node and hits the high endothelial venule to bring in monocytes. Now, another key observation that this paper made that I love so much is it's really the paper that pioneered our understanding of monocyte subsets. So the paper already needed to look at monocytes in the blood, and so used the CX3CR1 GFP knock-in that the Lippmann lab had made. And they already were beginning to understand in this paper that there were potential subsets of monocytes. So they acquired an antibody to CCR2, which were essentially not very common at the time. Rodrigo Bravo provided one, and you could see that there was only one population of GFP intermediate cells or GFP medium cells that was CCR2 positive, and then there was this other population. So this paper also led the way to really, precedes the paper that often in the field we think of as defining monocyte subsets. So I consider this paper to be particularly ahead of its time and very important for thinking about lymph node reactivity and inflammation, as well as monocyte biology. So thanks for listening.
Hello, and thank you for the opportunity to highlight a JEM paper that revolutionized immunology. My name is Stephanie Eisenbarth, and I'm at Yale University School of Medicine, and I'm here with current and former trainees from my group who will each introduce themselves and will introduce the paper that we chose.
Hi, I'm Sam Olyha. I'm an MD PhD student in Dr. Eisenbarth's lab, and today we're going to highlight the paper, The Identification of a Novel Cell Type in Peripheral Lymphoid Organs of Mice, Morphology, Quantification, and Tissue Distribution by Steinman and Cohn from 1973.
Hi, my name is Gowthaman Uthaman, and I'm now a PI at UMass Med School. I'm formerly from the Eisenbarth lab. So coming back to this paper, by the late 60s, it had become well known that for the induction of lymphocyte activation and antibody production in vitro were needed, in addition to T cells and B cells, a population of accessory cells that typically was found tightly stuck to the tissue culture plastic. Steinman, as a postdoc in Cohn's lab, wanted to study what these cells were. He characterized splenocytes that could attach to the tissue culture dishes and found a novel population of cells. These cells were often stellate with numerous projections or dendrites. After resisting the initial temptation to name the cells Claudiocytes after his wife, Claudia, he coined the term dendritic cells to describe them. He used differential staining techniques and electron microscopy to show they are distinct from lymphocytes, macrophages, and granulocytes. He noted that dendritic cells had far fewer lysosomes than macrophages, and these were perinuclear. His time-lapse images showed that dendritic cells' cytoplasmic projections were rapidly put out and withdrawn. He demonstrated that these cells were up to about 1% of cells in various lymphoid organs. As often true for most big discoveries in science, you see what everyone sees, but observe and think what others haven't. Steinman pushed dendritic cells. In subsequent years, his studies established that dendritic cells were critical for T cell activation, and the rest, as they say, is history.
So Ralph's landmark JEM paper set the stage for immunologists to understand the complex interactions between these professional antigen-presenting cells and naive T cells, and their role in shaping adaptive immune responses. Although DCs actually remain controversial and not well accepted for quite some time, ultimately the field grew with incredible tools developed by numerous investigators across the world now. The ensuing 40 years after this paper came out, the study of DCs revealed how the immune system differentiates and appropriately responds to different stimuli, from a strong antiviral response, T and B cell response, to a tolerogenic response to our own damaged self cells. DCs are the translators between the innate immune system and the adaptive immune system, and thereby act as gatekeepers for these very different types of immune reactions. For this, Ralph ultimately won the Nobel Prize in 2011. Ultimately, the identification of DCs and this first hint at their function had tremendous impact on immunology as a field, but has also been the foundation for much of the work in my own lab, aiming to understand how different types of DCs shape different types of adaptive immune responses. For this, I'm very thankful. And so a shout out to JEM for publishing this groundbreaking but controversial work at the time, and happy anniversary.
Happy anniversary.
Thank you.
Hello, everyone. My name is Ellebedy. I'm an associate professor of pathology immunology at Washington University School of Medicine. I'm delighted to join you in celebrating the 125th anniversary of the Journal of Experimental Medicine, a truly remarkable milestone. And I'm going to highlight a landmark paper that has been that was published 55 years ago in the Journal of Experimental Medicine. And at the time, this paper addressed a very important question. We knew by early 60s in the last century that secondary antibody responses are greater in magnitude and faster in kinetics compared to primary antibody responses. The immune cells responsible for that enhanced response was unclear. And as you can imagine, at the time there was no flow cytometry, so the cells that were mainly defined based on their morphological appearance. And there was a debate at the time that whether the immunological memory was carried by these large dividing lymphocytes that was generated during the primary immune response. And the dividing part was evidenced by the uptake of tritiated thymidine. Or is mediated by the small lymphocytes that we know now as memory cells that also generated during the primary immune response. So to address this question, the authors went for a very simple experimental model where they immunized rats subcutaneously with purified bacteriophages. And then after a period of rest, they took thoracic duct lymphocytes from those immunized rats and then adoptively transferred them into naive rats and then challenged the recipient rats. And you can see a very nice recall responses in 3 separate experiments here. But the problem with that transfer of this fresh thoracic duct cells, that you're actually having a mix of those large dividing lymphocytes and small lymphocytes. So which cells mediated this recall response? To address that, the authors used a very clever way to actually deplete the large dividing lymphocytes by incubating those cells at 37 degrees for 24 hours. And that led to complete death of those large dividing lymphocytes. And what remained is these small lymphocytes. So now the experiment can be comparing the recall responses from those immunized rats, in rats, if you take the cells and just inject them fresh in naive rats, or you actually take them and incubate them to get rid of those large dividing lymphocytes and then inject them and then look at the recall responses. And the authors did this experiment and the results were really clear that regardless now, even if incubated the cells completely, which completely got rid of the large lymphocytes, you have a really nice secondary response that was either equivalent or even higher in magnitude compared to if you just transfer fresh cells, leading the authors to conclude that this immunological memory is carried by the small lymphocytes, which we now know as memory cells. Thank you.